Hello, students, and welcome to my screencast on cellular communication. And in addition to this screencast, I'd encourage you to check out the Bozeman Science videos or Mr. Knufke's videos or Crash Course or anything that helps you understand the material. The important thing is not to memorize every single step of cellular communication. It gets quite complex, but to be able to just identify a couple of principles, right? Reception, transduction, and response, and then see how those interplays can happen with diseases like cancer, etc. All right, this is my first video this year, but I do a lot of them for the ninth graders, and all of them are licensed under non-commercial share alike, so I'm not making any money off of this. This is just meant to help you and help any students that might stumble across it. So let's get into the cellular communication items. You'll see a lot of pictures from your book and from Khan Academy, and those are available on khanacademy.org or OpenStax. All right, here are some questions that I got from Mr. Knufke, who I try and model some of the course after his good work. So can you pause the video and answer these questions? All right, I gave you a fair number of them in your reading guide, but it's always good to go back. Okay, how'd you do? Well, if you can answer all of them and you aced it, then you probably don't need to watch the video. But if you struggle with some of it or if you need a little bit of a refresher, here we go. All right, so cells are going to communicate. They might need to send signals to make other proteins or to send a message like between neurons, or a cell next to it might say, hey, let's grow into this space. So there's a lot of reasons that cells would communicate. A cool example might be like a macrophage presenting part of a virus to a helper T cell so that you can activate the antibody response. So cells don't just sit there on their own, right? They're talking to each other. So there's various forms of cellular communication. It's called your PJs, get it? Paracrine, endocrine, autocrine, juxtacrine, and synaptic. You don't need to memorize these terms. They're pretty self-explanatory. Auto meaning self. It's going to stimulate itself. In class, we looked at luciferase and looking at Vibrio fischeri and the bacteria that light up when they have enough autoinducer molecules around. Juxtacrine just means that they're touching each other. Paracrine would be nearby. Synaptic deals with neurons, and endocrine deals with your hormones. All right, and there's our little example. And then here is my daughter in her PJs thinking about cellular communication. Okay, here's just some examples, right? If you memorize something, it's not going to be as useful as if you see it in context and see it with an example. So here, autocrine, we talked about the Vibrio fischeri bacteria, maybe developmental cells as you're growing from a fertilized egg into a blastula, gastrula, etc. Cancer cells can autoinduce themselves. Here we talked a little bit about helper T cells that are touching each other and doing this, maybe through a gap junction. Paracrine, nearby, a good example of that would be the neurotransmitters. And endocrine, we talked a lot about insulin as a protein-based hormone that would go from your pancreas to cells throughout your body. So insulin can travel through the bloodstream. We also send sex hormones, of course, and other hormones that are lipid-based. They would need a carrier in the bloodstream because they are lipids. But once they get to the cell, they can diffuse easily in because they're nonpolar. All right, you can go through on Classroom. This is the exact same presentation on Classroom, and click on these and see some of the examples. And we looked at a YouTube video about bacterial quorum sensing and bobtail squid. I hope you found that cool. Here's another one. This would be with the antigen presentation by a immune cell, by a macrophage. Here is a good example of paracrine signaling. So maybe we need to clot a tear in a blood vessel, or we need to grow in a certain section. Here's some synaptic examples. This is kind of cool, dealing a little bit with autism. And so we talked about this with acetylcholine, right? Being released from a neurotransmitter to, say, a muscle cell, or maybe it's dopamine being released on a cell. We'll go over endocrine system in depth later in the year, and same with the nervous system as well. But here's a good example of that. So inter means between, so intercellular, between cells. And once they communicate or they send a ligand to each other, there's an intracellular response, right? And so that's what that AP question I put on your reading quiz was asking. It was pretty simple. It just said, hey, a ligand docks here. So that's the reception, right? Reception right here. Just like if you walked into TASM, you go to reception, then you get transduced or sent to where you're going to go. There's a lot in play right here. There's secondary messengers. There's the ability to amplify that signal. And then there's going to be some type of cellular response. A lot of times that could be expressing a certain gene, or it could be activating enzymes within the cell. And here it is. And you saw it was literally just like this on your AP free response release question. So it's pretty cool. Okay, I'd encourage you to go through that Bozeman science video right here that I've linked. It's right, obviously you would need to do it through Classroom, but he's got some animations that I just can't quite make yet. They go through a signal transduction pathway that are really showing the protein kinases being activated. So it's pretty cool. So here, I took this from Khan Academy. I just thought it was a good example, right? This would be like a G protein receptor. So it activates at the membrane. It's going to activate adenylyl cyclase, and it's going to make a secondary response. And here it comes, right? And it's going to send this on down. And it's hitting not just one protein at a time, but hitting many, oftentimes to make an amplified response. Here's a little picture of one. So this would be epidermal growth factor. Hitting a receptor, it's going to activate protein kinases in here. Whenever you see something get phosphorylated, it makes it do something, right? So it's going to make it active students. And so here it's phosphorylating, and then it's able to get into the nucleus through the nuclear pores, and it's able to transcribe a gene. And so this would be prompting cell division. Okay, we already went over this, but just this is a slide on it. So reception, transduction, and response. So once again, that's pretty easy to memorize, but you should try and see it with different examples. And I'll give you some more in class. We still have a couple of class meetings before the test. And so we're going to go over some some cellular communication diseases. Remember that I've posted, just to hold myself accountable, the schedule for the rest of the school year on there. Now, of course, we might have to make some adjustments, etc., when lab materials are available. And of course, to meet your readiness on certain items. And let's say you're acing something, we'll need to go ahead. Or if you're struggling, then maybe we need to stay behind on a topic. And that's just part of being a teacher and part of being a student. And we're in this journey together. And as I told you, my two big goals are to be a good dad and be a good AP bio teacher. So let's do it. All right. Here's the exact same picture. I don't know why I put that in again. All right, this is from Mr. Konofsky, but here's a good example of how universal it is. So this is kind of showing that this is a major area of evolution. It's something that's conserved, kind of like when we talk about cytochrome C and how we set up cellular respiration. It's something that's you see in a lot of organisms. So this is a way that cells have evolved to communicate. And so you do it, your dog does it, fish do it, etc. Here would be an example of different receptor proteins. So I told you pretty clearly in your study guide and in your reading guide that you don't need to know the different ones. And where I get that information from is the curriculum framework guideline on the AP biology website. You can always check that out. It's there. It's a lot of pages long. It's one of the things we have to go through as AP teachers to get certified to teach this. All right, so here, this is pretty cool, right? You're seeing an adrenaline molecule come in, epinephrine, so you're getting ready to run away or fight or do something, and it's going to activate this receptor. It then is going to catalyze this adenylyl cyclase, which is going to send out a bunch of cyclic AMP. Notice how here's one molecule, and here's 1, 2, 3, 4, 5. So we're already seeing an amplification effect, and then they're going to activate other molecules. Pretty cool. So here's just an example of it, right? I put a bonus question on your reading quiz about that. I think it was more about the receptor complex up here, but hopefully you could see from that released AP question that you don't need to know each individual molecule, et cetera. That would be insane to try and memorize that. I wouldn't be able to do it. I doubt many biology graduates would do that either. The point is, if you start studying a certain pathway, let's say that you're going to do a big project on insulin and insulin receptivity and what's going on with that GLUT4 transporter and diabetes, then you would get into those individual molecules. All right, and so here, this is just showing how complex they can be. And so you might say, well, why don't you just hit one receptor and then bam, you get the response. And the reason it hasn't evolved that way is this gives you a lot of complex methods of control and gene expression and amplification as well. It's kind of akin to your phone, right? If you look at phones nowadays, they're super complex, but you can do so much stuff with them. In the old days, like me, when I got a phone in 1999 or 2000 or something, you press one button. Right? But now you can have all these different interplays, which give you complexity. It's emergent properties at play here. Okay, here we go. So here's the amplification one that we talked about. So this is a classic example. So here's adrenaline binding to a receptor, and then you're going to get 10 to the 6th enzymes, glycogen phosphorylase, which is going to break down glycogen into glucose so that you can go use it in your body. We went over this pretty clearly in class. Right? If something is nonpolar, if it's derived from cholesterol, it's going to be able to go right through the membrane, whereas if it's polar, it's going to have to dock with a receptor. So sometimes those receptors can have mutations in them, right? And then it won't work anymore. So here's, I asked this question on the reading quiz. Not many people chose to do it, which means at OpenStax just didn't really go into it. But here, as much as I would have liked, here you have the same molecule or messenger, and it's hitting different cells, right? It's hitting one in your intestine, one in your muscles, and one in your liver, and it's going to give different responses. So they all have receptors for it, but they're going to have different responses on the inside, which is pretty cool, right? One molecule or one signal can lead to different responses. So here the vessel constricts, right? It's pushing blood away from your intestines. And where is it going to? It's going to your muscles so that it can carry that glucose. So here the vessel dilates or gets larger. And in your liver cell, it's going to activate that glycogen phosphorylase to break it down into glucose. So one signal, different effects in different cells. It's pretty cool. Another example of regulation and emergent properties. Okay, I hope you enjoyed this first one. I'm going to try and do another one or two before DO, and I'm just plugging away. And then if not, I'll be able to finish them after DO and post in the classroom before that weekend starts. Take care and have a great trip if you're watching this early. And if not, I hope you had a great one when I see you back in class. Take care, students.